Okay, now you have to do a conversion, right, to get it to millimeters. Okay, so how are we going to do that? So I'll flip you guys to show you the board. How are we going to do that in, how are we going to do that? What do we have, 2 centimeters, right? 2 centimeters?
Yeah, 2 centimeters.
Okay, and we want it in what?
To millimeters.
Millimeters. All right, so how are we going to do that conversion?
You times centimeter 2. Oh, you want to go?
No, go ahead.
You times it, and then after that you divide it. So, I believe one centimeter going to millimeter, 1,000, or 100.
How many, you guys just counted it on your ruler. How many little lines were in 1 centimeter, right? How many millimeters are in 1 centimeter? Just 1 centimeter on there. How many little lines?
I thought 10.
Yeah, 10, right? There's 10 millimeters in 1 centimeter. So to do this conversion, you can do it 2 ways. Remember we were talking about the decimal place moving,
Yeah.
right? So if we know that this is a smaller unit and this is a larger unit, we're going to move our decimal place to the right, okay? So how many places to the right if it's 10? Yeah, just 1, right? So then what's our answer? 20, right? You also can write it out. So if the decimal place thing throws you off, right? You can kind of do it like this. You could say 2 centimeters, right? And then you know you want centimeters to cancel out. How many millimeters are in a centimeter? Right? So it's just times, 2 times 10, right? Okay? So that's an easy conversion. Right
It's not
now,
letting me type in here.
huh?
It's not letting me type.
Well, you have to enable editing or something like that. It's a Word document. All right, so does everyone see how to do that conversion? Yeah? Okay, so now what about what I asked you next? Oh, what's the length in meters now? So you can either start with 2 centimeters or 20 millimeters, it doesn't really matter. So if we go with 2 centimeters, now we want to know meters, right? So it's different in a couple ways, right? Meters, is that a larger unit or a smaller?
Larger.
Larger, right? Probably hard to see the red, I just realized, but 2 centimeters is larger. I mean, meters is larger than centimeters, right? So then which way are we gonna move our decimal place?
Left.
Left, okay, good. Now, how many centimeters are in a meter?
100.
100. So how many decimal places do we move it to the left?
2.
Correct, 2. So then what do we end up with? 0.02, right? You could also, again, you could write it out, right? So if you wrote it out, you could do 2 centimeters, right? And then again, you got your, we want it in meters, right? So 1 meter, there's 100 centimeters, right? So it's 2 divided by 100. Same thing, okay?
Question.
Yeah.
In this class, do we do significant figures or no?
You don't have to worry about significant figures. Yeah, don't worry about that. You can save that for chemistry or whatever.
All right. Okay.
I just want you guys to know how to do it. Yeah, so Erica asked about significant figures. You don't have to worry about that, just as long as you get the right answer, okay?
Okay.
Yeah? Okay, so we did that conversion. So now we're moving on here. So we move on. Oh, it says number 3. It says describe how you did the conversion, right? So you got to write out, I don't know. You can even just write out like the equation like that, or just say I moved the decimal place, 2 spots to the left, something like that, okay?
Okay.
All right, so take a second to write down how you did that, right?
You want us to write it in words too or no?
No, you don't have to write in words. You can just kind of show your equation, that's fine. If you're handwriting it, it's easier. All right, you guys on Zoom, you have questions or you're all right?
I'm good.
Good? Okay. All right, so we move on to number 4 there. So you guys have a beaker and you have a graduated cylinder in front of you there, right? So we're here. You guys can, oh yeah, I can use that as my point of view there, right? So you have a graduated cylinder, you have a beaker. So both you guys, the first question I think asked you about the graduated cylinder, right? What's the volume, okay? So basically we're just gonna measure the volume in that graduated cylinder. Actually, I don't even need to take this off. So how do you measure the volume in that graduated cylinder?
Imagine by the water that's going down.
Yeah, the bottom of the meniscus.
Yeah.
Good. So you have to get eye level, right?
Hold on.
And then write down how much volume you have in there.
I gotta move it a little closer.
Yeah, that's fine. Yeah, you can move it.
I need to get me some new glasses. If I'm not mistaken, it's like 28. So it's going to 29, I believe.
Okay, yeah, it looks like that. Yeah, I would say 28. All right, so 28 what? What's yours there, Sting?
Milliliters?
Yep,
22, is it?
yeah, 22. Yep. So 22 milliliters,
So
right?
when we put 22, I just put 28.
Oh, yeah, no, you put 28.
All
You
right.
put what yours is. So Sting's was 22 milliliters and Erica's was,
28.
what the hell she called me for? Sorry. It was 28 milliliters. All right, cool. So again, it's another conversion, right? So 28 milliliters, right? What is that? Let me switch to a different color. The red does not show up very well. 28 milliliters. Now I'm asking you guys what that is in liters. I might be able to see from this. Okay. All right, anyway. I got too many cameras going here. Let's push this up a little bit. Okay, so what do we have? We'll go with 28. It doesn't really matter, 28 or 22. You guys can figure that out, right? So 28, I can write that, 28 milliliters. And we want to know how many liters that is. Can you kind of see that a little better? Maybe. So what do you got? What are we going to do? Or what do we need to know? What's kind of our first thing that we need to know?
How many milliliters?
Yeah.
Good. How many milliliters are in a liter? How many?
1 liter, oh, it's 1,000 milliliters in 1 liter.
Good. 1,000 milliliters in 1 liter. Perfect. Okay. So now remember, this is our larger unit, right? So if we're talking about decimal places, which way are we going to move it?
Left.
Good. To the left. The number's going to get smaller. So how many spots to the left if it's 1,000? 3. Yep. Right? So then what do we end up with for our answer? Point. Go ahead. What do you say? What is 0.028, right? Because you got to go 3. Your decimal place was here, 1, 2, and then you got to add another 0 there.
Oh,
3,
okay.
right?
Yeah.
You got that? Erica, you got that?
Yes, ma'am.
All right. And so again, if you wanted to do your division or whatever, you could do that too, right? So 28 milliliters. Okay, we need liters. We put milliliters down here. We know there's 1,000 milliliters, right, in 1 liter. So it's 28 divided by 1,000, okay? Or 0.028, 0.022, depending on what you started with. Questions on Zoom, guys? No? Okay. All right. Now it asks you to measure the amount of water in the beaker. We'll swing back to Erica here. Amount of water in the beaker. So there's your beaker. Yeah, how much you got there? Estimate the best you can.
And 27.
All right. Write that down. What do you got there? Yeah, so what do you think?
By 50.
So this is 150, so then that line, what do you think that line is there?
140? No. I don't know.
Less than 140.
130?
Well, so it's exactly halfway between 100 and 150, right? So 125 to that line, right? This is going 25, 50, 75, 100, 125,
Oh.
okay? All right, so 125 milliliters in the beaker. Do the conversion. Tell me what the answer is. So now we're starting with what, 125 or 127? The same thing we just did, right?
All right. I have received 0.127.
Yeah, 0.127, right? Yeah, correct. Sting, did you get that?
So you don't add the 0?
Why not? Because look, we're starting here, remember? And how many decimal places is it?
3.
3, right? So if we move it 3, 1, 2, 3, right? Oh, the only time you're adding a 0 is if you had to go like another one, right? So remember last time we started with 27 milliliters, right? And we had to go 3, so then it was 1, 2, and we had to add the extra one,
Oh, okay.
right? Does that make sense?
Yes.
Okay. Right, so the answer is 0.127, right? If you want to add a 0 in front, you can, but 0.127, yeah? All right. Okay, I didn't put this on here, but you can add it in. What is that? So 127 milliliters in microliters. So now, which one's smaller, microliters or milliliters?
Microliter.
It's smaller, right? And by what factor? By how much? What do you think? When in doubt, what should you choose?
So that I'm sorry.
How different, what factor, like, is this different, right? Microliters, or how many microliters are there in a milliliter? 1,000. Yeah, good, okay? But now this is a smaller unit, right? So which way are we gonna move our decimal place?
To the left. Oh.
To the right, right? This number, so
Oh,
remember,
way too small.
right, if this is smaller, right, you're gonna end up having a lot more of these, right? And so if we add 3 to the right, what do we end up with? Just adding 3 zeros on, yeah?
Yeah, so it'll be 1.
127,000, okay, microliters in 127 milliliters, right? So in this amount of liquid here, of water, right, there are 127,000 microliters in 127 milliliters, okay? Now, usually you're not, like, you wouldn't do that conversion probably, but like if you have a smaller amount of like milliliters, you may need to do that in the lab, go back to microliters, okay? You guys good over there? On Zoom, yeah? Okay. All right, so add in that conversion. You should kind of write that one in, how much it is in microliters, okay? For number 5, right? All right. You good?
Yep.
Okay, so we move on. So now we're gonna move on to the microscope, okay? So what I want you guys to do is just uncover your microscope, turn it on, get the light on, and we'll go from there, okay? And I actually can put, I'll show you.
You want us to take this off now?
Yeah, go ahead. You can take it off. Yep, take that off. You can turn on the microscope. And it's not a letter A slide, it's a letter E slide, but anyway, we're not gonna put it in just yet anyway, okay? So let me unshare that because I'm not showing anything. If I want to show you guys something under my microscope, I can show you it that way. Okay, so what you want to do first, guys, with the microscope, right, is you want to, I'm going to just put it here so you can see your microscope well. Okay, perfect. And Sting's in there too. All right, cool. So you want to turn this here, right? This is how you switch the objective lens so that the scanning one is in the middle, clicks in the middle. Okay, so turn it so you get the scanning one in there. So that's the lowest power one you have on there.
Yes.
Usually it's red. All right, there you go. Click it in. It'll click in. Got it? Clicked in there? Yeah? Okay. So then I asked you, what's the magnification of that scanning objective? So take a look at it. Sometimes you can see it like actually on the side of the objective. It's labeled.
So this one is kind of erased, but I think this 43. This 43? No, it's 4, right?
4. Yep, it's 4. Okay. So the magnification of this lens, this scanning objective lens, is 4x. All right. Now you're going to take your letter E slide. It's not a letter A. I don't know why I wrote letter A on there, but anyway. Okay, and we're going to place it on the stage here. So you're gonna have, yeah, you pull the one side, you got it. Slide it all the way back. All right, cool. All right, now we want to get that letter E in focus, okay? So under this scanning power, I'll move this so we don't knock it over. So that was me. I've already probably knocked it over. All right, so now you want to get that in focus.
One question.
Yeah.
Can I open it?
So, okay, you can. So for the ocular lens, it actually, you don't even have to look in both at once. If it's more comfortable just looking in one, you can just
Yeah,
look
that's
in
one
one.
that actually.
That's what I do.
Like,
Yeah. All right, so now here, to get this in here, you hold this guy, right? And then slide it to the back there and it's in. Now to move yeah, you got it, Erica. To move this around, right, to get it in the middle, you move it this way. Oh, turn your, over there, I think the on. Yeah, see if you need to, oh, you're not plugged in. What the hell? Where'd your plug go? That's interesting. Okay. Anyway, we'll switch up. I think you can pull your slide out of there and put it in this one. All right, so get it in focus under scanning. That's your next thing to do there. You got it. Slide it all the way back. Okay, so then that part goes in the corner there. Yeah, you got it. Let me move this out of the way. You can slide that over if you want.
I thought I found it. That's my eyelash.
Yeah, sometimes your eyelashes get in the way. That's true.
I'm like, I found it.
All right, good. You're good. Slide it all the way back there like that. Now, this will turn it on. This is your light intensity here. So once you get it on, then you're going to use this, right, to move it so that you get your E right in the middle there. Okay? And then also flip it so your scanning objective is in there. Did you get it in focus? I'm going to put one on my microscope. Worked yesterday. Oh, maybe it's working. All right. So you guys see my screen. You can actually see. This is what I have in my microscope. It's sort of uninteresting. Obviously, the letter E. All right. Right. So we want to get it in focus.
How you found it that fast? You found yours?
So that's what you should see, right? A little lowercase e.
Yeah, but how you
Well,
found
big lowercase e, I guess, when you look at the microscope.
Not that fast.
So the best thing to kind of do when you're trying to get it in focus is to start off with turn it this. Here, I'll show you. Okay, so from here. Start from here, then look in there, and then slowly turn it like this, and you'll get it into focus. Now remember, you may have to kind of like move the location, but I think you're kind of in the middle there, so you should be okay. All right, so slowly turn it towards you. Yep, and it'll come into focus. You got it in the middle here. Look. So get it in the middle. All right. Okay. Now I'm gonna start you here. Okay. Now look in there and then turn it this way slowly until it comes in focus. Okay. That big knob here. This guy. That's the coarse adjustment knob. So you look and you turn it slowly, and it should come into focus for you. Did you get it? You got it? Oh, you got your eyelash?
I want to make sure. Did I put it on there? Go on, did I keep popping up my eyelash? So I think I need to comb them up.
Yeah, you have to kind of get used to that. I mean, this is why, right? You have to get used to using this. It's not something that you just watch somebody use and then know how to use it. You don't. Got it? So keep going. You probably have to get closer with this. If you feel like, do you see it a little bit? You might kind of start to see like a little blurry kind of black thing, which eventually it'll come into focus.
It looked like it's focused on here. Like when I look like this, when I look.
Well, yeah, because you're not, I mean, you mean it looks like it's in the middle,
Yeah.
but it's just not in focus, meaning it has to get closer or further away, right? So that it, but it could be slightly off to the side too. Did you get it, Steve? If you want, I'll help you.
I don't want to try a little bit more. I
All
wanna
right,
get
yeah,
it
go
right.
ahead. You just let me know.
Yeah, I'm gonna need your help.
That's right. All right. So let me see here. Oh, you know what? Oh my God, with the mask, it's hard to see what the hell is going on here. Something's not right with this thing, by the way. So probably why you're having trouble. Oh. I don't see anything. We have to move. Give you another microscope. Let's see. What the hell? Oh, there it is. What in the heck? Oh, here. Yes, this is closed. That's what it was. Right here. Now
Oh.
look, I'll show you. So it was there. So that was closed, right? So then the light coming through was just this tiny little pinhole, and then that's why you couldn't focus.
Oh,
So
okay.
there you go.
I should have taken that on.
Yeah, but then so I don't know why that got changed. Did you get it?
No.
With the way the light's coming in. Oh, there it is. See it.
Oh, okay.
Yeah, sure. All right. So now, one thing to look at like what if you look at your slide there. So I think yours, like if you look at this with the naked eye, it's hard to see, but you can see how your E is like upside down. But when you look at it under here, what does it look like? Now, Erica, yours is the opposite, right? So yours, you put sort of right side. Like if you looked at this just with your naked eye, it's went right side. But when you look under there, it's inverted. And if you guys look at my screen, you can see that my letter E is upside down, right? So
Mine's upside down too.
the microscope inverts the image. The image is always inverted. All right? All right, so draw what you see under that scanning, what you got right now. Okay, now you're gonna change. So now it's in focus. So whenever you're focusing something on the microscope, like any slide, and I have another slide I'll show you guys. But you always want to start under scanning like we did, okay? Get it in focus. Now you want to get it under higher magnification. So now you're going to switch your objective. Don't touch anything else. Just switch your objective to the low power one. Usually it's the yellow one. Yep, okay? And now I ask you what the magnification is of that objective. What is it? Do you know? It's same thing. If you look at the side, it probably will tell you somewhere.
I still see even a little more blurry.
Yeah, so then yeah, I'll show you. So like look, so now it's 10x. The low power objective is 10x. So if I switch it, look, right? And mine's a little bit, I think the field of view a little different, but anyway. Right now you can see part of the E, but it's much more magnified, right? In yours, I think usually it's just because of the way that camera is. You probably can see the whole E, but it's bigger.
Yeah.
Yeah? Okay, now, if it's slightly out of focus at this point, all you need to do now, you don't want to use this coarse adjustment, the big knob, you'll use the small knob. So just ever so slightly adjust it using that smaller knob and it'll come into better focus. Yep. So do the same thing, just, I mean, unless it's completely in focus, but you can use this little knob just to get it in better focus. It's already completely in focus, like it should be pretty crisp, like right and clear. If it looks a little blurry, that means you want to turn that little knob just to get it in a little bit better focus. Yep,
Yeah, I
you
see.
got it. Right, if you turn it, you'll see it'll become more clear. Yeah? All right, cool. So draw what you see there, under B there.
So you just want to draw like a big.
You're just going to draw a bigger E, yeah. Right, so that E is magnified. If you guys are following along, you can draw what you see on my screen on Zoom there, okay? The E gets bigger. Because why? Now you're using a 10x objective here, right? Okay, before we were at 4x, now we're at 10x. Now you're going to switch up one more, okay? So to the high power objective, it's usually blue. I think it's blue. Yep. All right, you got it in there? Now, same thing. It should just about be in focus. You may have to just use that fine adjustment just to get it clear. Now, you're not going to see the whole E, right? Because now we magnified it even more. So you're probably just going to see like one little black line or something like that, like a piece of whatever. You're not going to even know what the heck it is, right? You got it? So now, yeah, so now you got to be careful, right? So when it's under high power, it's harder to focus, right? So that's why I say you always want to get under low power. And then once you get here, you usually want to just adjust this a little bit. Otherwise, then you like completely lose it and it's hard to get back. So see if you can get it back. Otherwise, I'll help you. Otherwise, you know what you can do? You can always go back
to focus
to the lower objective, get it in focus, and then switch back again. That's the easiest way to kind of do it. Did you get it? See? Just probably like a piece of the E, you see. Let me see. Oh, it kind of went out of view. So here, look, I'm going to put it in view. All right, so now if you look, there's like a gray blurry thing. So use the fine adjustment there, that little knob, and get it into clear focus.
Yeah, mine's just totally just went away.
Oh, it's gone?
Yeah.
All right, so if you guys look on mine, I'm going to have to increase the light. Look, if you just look over there, right, you see I have like one little piece of it there. I don't see that. Right, that black stuff on the side, that's just my little piece of E. That's the only thing I can see now because it's so magnified, right? The field of view gets smaller when you go under higher magnification and you're making that image bigger. So you can't see the whole E anymore, right? Because now you zoomed in on one little part of the E. Make sense? Okay, so you're just gonna probably just draw like a kind of like a little, I don't know, some black shading on one side or something like that. And then write in what the magnification is. Did you get it or you completely lost it? Yeah, so then best, you can even just go all the way back here, right? Get it back in focus. I see it there, I think. There it is. Yeah, it just got out of focus. And then you can switch back.
All right, if I'm not mistaken, blue is 40, right?
Correct. Blue is 40. Yep. The high power objective. All right, so now look here. It's out of focus, but you can use this to adjust it to get it in focus. All right? See that? You kind of just see a blurry kind of black. So now you use the fine adjustment.
I just use the small one,
right? Then a small knob. And you'll be able to get in focus. Sometimes you'll be able to tell you're going the wrong way, then you have to turn the other way. You got it?
Yes.
All right. Okay, part D, answer that one. What's the total magnification when you're using your microscope under high power? What do we mean by total magnification? I went through this in the lab demo.
Wait, say that again? Sorry.
Total magnification.
Like the highest one?
Well, the term total magnification, right? It encompasses both the ocular lens magnification and the objective you're using. So right now we're using 40x here, right? But how much are the ocular lenses worth? It actually says it on the side, I think, of yours.
10.
10, right? So the total magnification is the ocular 10 times whatever objective you're using.
Yeah.
So what is it?
400.
Yep, 10 times 40, 400. Good. All right.
You put that right here?
Yeah, just part D there. See that? What's the total magnification when you're using your microscope under high power? It's 400x.
I think your mic was muted.
Oh, yeah, but I think this one's on, is it? Let's make sure this, oh. What the hell did I do? Thank you. Oh, no, you know what? This one's not. This one's on. I have it on 2 screens. Thanks. I was like, oh, no. And nobody told me. That means nobody's paying attention. All right, you good? Erica, you see those were all those questions we went B, C,
Yeah.
D. Yeah. Okay. All right. Now we're on number 5. And by the way, oh no, we're gonna skip number 5. We're not gonna make cheek cells because I realize maybe because of COVID we shouldn't be taking
swiping.
toothpicks in our mouth and then putting it on a slide and whatever. So we're just gonna do 6 onion. Okay.
All right.
So you guys have a slide here. I'm gonna get you a little piece of onion. So we're gonna make a slide of onion tissue and look at some onion cells.
We're gonna
Okay.
use this one, right?
Blank slide. Yep. And then you're gonna need these cover slips. So first, just start with that slide, and I'll get you guys these onions. All right. We're trying to get you as thin as you. And this is the last thing we're doing. Try this. That's how it goes. Right. So, just gonna take a little piece of onion. Where's your slide? Okay, here. Okay, little piece of onion. All right, and then what you're gonna do is you're gonna take, you have iodine there, Erica, that's fine to use, and you have methylene blue thing, so you're just gonna take this dropper, take some of that methylene blue, drop it on there, a couple drops, and then you're gonna put one of these cover slips on top, okay? And then you're gonna get it in focus under scanning first. Here's your onion.
Right here.
Yep. All right, and then some drops of the iodine, cover slip on top, those little cover slips in that box, go on top after you put your iodine. Yep. So put enough so that like you feel like it changes color slightly.
Yeah, I think that's enough.
I'll make one too. So this is gonna be like a little harder to focus, just because you have like it's kind of like uneven. I didn't section it very well, because I'm just using a razor blade or whatever.
Is this enough color, or should I get a little more? It changed a little color.
Yeah, put a little more. Put a couple more drops on there. Yeah, you probably can put a little bit more here. There you go. All right, and then you just put your cover slip on top.
That's enough.
You kind of squeeze it down a little bit, but not really too hard because you'll break the cover slip, so be careful. Yep. Yep, there you go. So you can kind of push, you can push down a little bit like on it, and then you're gonna just put it under there. Yeah, so you'll see it's a little thick, so it's okay. You'll do the best you can with getting it in focus. And you want to get it in focus under scanning, just like we do. We start from the lowest power always, okay? And then we'll just go up to the low power. I think I asked you to draw what you see under. Oh. Anyway, you're going to draw what you see under 10x, under that low power, okay? Now, the stain is not very good, by the way. I know kind of seeing something like this. Oh, let me go get my lab manual real quick. There's a picture in there. So do you kind of have something that looks like that a little bit?
How you zoom in with this, right? If I'm not mistaken.
Oh yeah, you do. Thing. So you just get it a little more better in focus. Kind of if you get to one, yeah, yours looks pretty good actually. Then go one more up though. Get it in focus under low power. Okay. You got it?
No, that's not. I'm trying to zoom in.
So it is going to be kind of like hard because it's like uneven. So like sometimes you have to kind of like move it around to find like where the thinnest part is and just get it in focus the best you can. I'm going to find I have this other slide. I'll show you that. Then make it better. Oh, it was here. Did Mo move it? Let you guys get this.
All right, found some. All right.
Kind of got it. It's that, like I said, it's going to kind of look. Let me go get my lab manual so I can show you guys the picture that's in there. Okay. Did you get it in, switch to the 10x and try to get in there. So it's gonna kind of look something like this picture here.
Did this look good or should I?
Yeah, I think that's fine. I don't think you're gonna get better than that. Like I said, this isn't the best stain, right? It's not really that dark. So if you look in your lab manual, right, it kind of looks something like that, right? You guys, these are like tightly packed cells. And you can kind of see, if you zoom in enough, you might be able to see the nuclei, like you can see here.
Yeah, just the small circles.
Yeah. And then if we were going to do cheek cells, right, the cheek cells are a lot different shapes, right? They're more irregularly shaped, they're round, they're more spaced out,
Yeah.
right? But you can, same thing, you can see the nuclei. So like, if I showed you a picture like this, like when I give you guys a lab practical, you should be able to identify the nucleus, right? From these cells, and we should be able to like know that these are cheek cells and these are onion cells, right? Okay, so right, the onion cells and then versus the cheek cells. So if we did our slide of the cheek cells, we would be able to see that. But let me show you one other thing. I'm just gonna put this. What did I do with it? Oh, I wrote it over here. There's just a slide that was laying around of like a cerebellum. So it's the back part of your brain, the cerebellum, and so what you can do is you can take out like a mouse or a rat brain and you can section it very thin and then you can stain it, right? So see how this is like super dark stain? So our stain was stinky. The reason I want to show you this is that this is like a good stain and you'll see how you can actually see all the individual cells in the tissue, right? So let me just show you up here. It's more interesting to look at than the letter A. Right, so like each one of those like small purple dots that you see there, those are cells, right? And so you can see how they're like darkly stained. I'm gonna go one more. But so anyway, that's what a stain is supposed to do. It's supposed to actually like let you clearly see the cells. Okay? See? You can see all those individual cells there. All right, so I just wanted to kind of show you guys that. So that's just, those are just individual cells in the brain that you can see. All right, so you guys are done. You completed everything.
Yeah, I'm gonna try my best to draw this.
Yeah, just draw for the onion cells. It kind of, yeah, just draw the best you can. And it's not really about like accuracy, right? It's just proof that you guys did it pretty much. All right, so you guys that are on Zoom, right? They're just drawing what they see for the onion cells, we're not doing the cheek cells, okay? What I'm showing you now is just the picture of a cerebellum. So it's a slide of cerebellum, part of the brain. So let me put my onion back in here. It's pretty, not very good, but doesn't really matter. We can get the idea here. Let me see. 1 second, I'm gonna get it in focus for you guys. Maybe. All right, so anyway, just kind of do the best to just draw kind of what you see here. There's also a picture in the lab manual, or if you look up onion cells, you can see what they look like. They're kind of rigid and really tightly packed, okay? That's fine, yeah, that looks good, yep. All right, so this is the picture that's in the lab manual, if you can even see it, but it's also in your PDF, in the PDF of the lab that you guys did, okay? That's there, it's on page 24 of that, okay? Of that PDF. All right, guys, so those of you guys on Zoom, I think you probably got all the information you need, yeah? So you guys just need to submit that electronically, okay?
Okay.
All right, guys, I'm gonna stop the recording. I don't